Hello everybody, my name is María José Díez and in this video I will talk about how to phenotype tomato leaves. The tomato plant has compound leaves. A compound leaf is made up of leaflets which are distributed along the leaf rachis. While the entire leaf is connected to the stem by the petiole, the leaflets are connected to the rachis of the leaf by the petiolule. Some of the leaflets on this leaf are compound as well. Besides of the leaflets, there are also intercalary leaflets, similar to the leaflets but smaller are non-compounds. The complexity of tomato leaves is a plastic trait dependent on growth conditions and cultivar background. However, most importantly, the complexity depends on the position of a leaf along the shoot axis, where complexity increases acropetally from the first leaf to the penultimate leaf of the primary shoot. Aerial plant architecture is predominantly determined by shoot branching and leaf morphology. In this video, we will focus in the following leaf traits: leaf attitude, leaf length and width, size of leaflets, type of blade, intensity of green color, glossiness, blistering, and attitude of petiole of leaflet in relation to main axis. Any of them will be considered in the phenotyping trial of the repository of Harmston. But as we will see, all of them are included in the protocol for test of distinctness, uniformity, and stability of the Community Plant Variety Office. One of the traits included in the technical protocol for tomato is the leaf attitude. The attitude of the leaves with respect to the main stem vary according to the position in the stem. Young leaves in the upper part of the stem are usually erect, while old leaves are usually dropping. For this reason, the attitude of the leaves should be observed in the middle third part of the plant. You can observe here the different position of the leaves in respect of the stem. The trait erect attitude is very interesting because it facilitates the aeration of the leaves, preventing in this way the infection by fungi, especially in wide environments or after some periods of rain in greenhouses. In fact, common growing practices consist on removing the old basal leaves of the plant to prevent the proliferation of fungi. The position of the leaf can also be related with the sunlight received by the leaf and consequently with the photosynthetic efficiency. The area of the leaf is determined by its length, width, and the size of the leaflets. It can vary greatly between varieties. Leaves can be short, medium, or long. This trait should be measured from the tip of the terminal leaflet to the point of insertion of the petiole into the stem, and in a leaf completely developed. The width of the leaf should be measured as the maximum width in an expanded leaf. Both traits, length and width, are important to determine the size of the leaf. As you can see in these pictures, the ratio length-width can be very different. In the leaf of the left side, this ratio equals to 1.8. In the middle one, equals to 1.3. And in the leaf of the right side is 0.8. The size of leaflet should be observed in the middle of the leaf, because as you can see, not all the leaflets have the same size. They can be scored as very small, small, medium, large, and very large, by comparison with some standard varieties. Both traits, length and width of the leaves and size of the leaflets, influence the leaf area. Leaves are the most important organ for photosynthesis process in which light energy is apprehended by green plants and used to produce carbon compounds from carbon dioxide and water. There is a close relationship between leaf area and the solar radiation interception, which is associated with photosynthesis and transpiration processes, both of which are strongly linked to biomass accumulation and productivity. Another important aspect related with leaf size is the protection of the fruits against sun. Tomatoes love sun, but too much sun can produce sun scald. Sun scald is a physiological disorder that turns the tomato pale white or blotchy yellow due to the intense heat and sunlight. The plants on the left have a scarce foliar cover and the fruits are unprotected. However, the leaves on the right images are vigorous, big and wide. They cover the fruits and protect them from the sun. For this reason, the leaf area is an important parameter as it reflects the photosynthetic capability of the crop, which in turn influence the productivity and protect the fruits. Several methods and software for measuring leaf area are available for scientific experiments. The management of this software is easy and provides a very valuable information about the main traits of leaves. The leaf blade can be pinnate, bipinnate, and double feather. In pinnate leaves, primary leaflets do not bear secondary leaflets. In bipinnate leaves, primary leaflets again are pinnate, so they bear secondary leaflets. And there is still one more complicated type of leaves, double feather. In these leaves, secondary leaflets again are pinnate. This last category is not included in the technical protocol, but we consider it can be recorded if we have this type of leaf in our phenotyping trials. The gene BAP regulates leaf complexity. Another key gene regulator of leaf dissection is the C potato leaf. C mutants show reduced leaf complexity compared with the wild type, with 70 to 80% fewer leaflets, and the leaf blade margins are smooth, lacking lobes and serrations. Environmental factors such as light intensity influences greatly the color of the leaves. There are options to measure this variation: light, medium, and dark. The color of the leaves influences the photosynthetic efficiency. Normally, the color is darker when plants are grown in the open air or in areas with high solar irradiation. The glossiness can be weak or strong and should be observed in the upper side of the leaves in the middle of the plant. Blistering is the difference in height of the surface of the leaf between the veins. The blistering should be observed in the middle third of the plant and can be weak, medium, or strong. If spores of fungus are deposited in a strong blister leaf, probably it will be easier the germination of the spore and the initiation of an infection process. Finally, the attitude of the petiole of leaflet in relation to main axis can be semi-erect, horizontal, or semi-dropping. A leaflet in dropping attitude can difficult the deposition of spore of fungi on the surface of the leaves and consequently contribute to avoid infection by fungi. The opposite occurs in semi-erect leaflets. As we have seen, the leaves play an important role in the development of the plants, the protection of the fruits, the efficiency of the photosynthesis, and the establishment of infection by fungus or insects. We have seen some aspects related to leaf morphology, but the variation is much bigger than the images I have shown you. Here you have some more photographs to illustrate the extraordinary variation you can find in the leaves of the cultivated tomato. Thank you for your attention. I hope this video has been useful for you.